Hello everyone, this is our lecture number 4 of bacterial structure. In this lecture, we are going to understand about cell wall. Let's start our lecture. First, we are going to understand the introduction. Below this external structure, capsules, sheaths, flagella, and above the cytoplasmic membrane is the cell wall. This is a very rigid structure and provides definite shape to the cell. Now see, the cell wall is present above the cell membrane and different body parts are external to this structure. And this is a very rigid structure and it provides definite shape to the cell wall. Since most of the bacteria lies in the hypotonic environment, this cell wall prevents the cell from expanding and eventually bursting because of uptake of water. The cell wall is resistant to extremely high pressure. Now we know that cell wall is the external structure to the cell. That's what it provides the cell from the external environment. For example, high temperature, high pH and high pressure also. The cell wall constitutes significantly portion of the dry weight of the cell. It may account for such as 10 to 40% dry weight of the bacterial cell. Bacterial cell walls are usually essential for the growth and division of bacteria. Generally, cell wall is made up of large number of layers. The thickness of this bacterial different layers varies in both gram-negative and gram-positive bacterial cell. The walls of gram-positive and gram-negative are different from each other. Mainly, gram-negative species are generally thinner, that is 10 to 15 nanometer, while gram-positive species are 20 to 25 nanometer in nature. Let's see next point, that is the chemical composition of cell wall. The most important constituent of the cell wall of eubacteria is the peptidoglycan, which is sometimes called as a murin, which is an insoluble, porous, cross-linked polymer of enormous length and rigidity. Now we can see here the peptidoglycan structure. This peptidoglycan is only found in prokaryotes. It is basically polymer of N-acetyl glucosamine, which is called as NAG, and N-acetylmuramic acid, which is called as NAM, and 4 amino acids, namely L-alanine, D-alanine, D-glutamate and D-amino acids. The tetrapeptide of one peptidoglycan layer is cross-linked with another peptidoglycan layer. As a result, strong framework is formed around the cell and impart a great rigidity to the total structure. We can see these different layers of peptidoglycan which are linked with each other with the help of linkage, and this will helpful for the rigidity of structure. Now here also we can observe the gram positive and gram negative cell wall. Now here peptidoglycan layer is very thick in the gram positive, where it is very thin in the gram negative. Periplasmic space is present here at only one region, where in the gram-negative bacterial cell, periplasmic place present at 2 space. Plasma membrane is very thin in the gram-positive, which is very thick in the gram-negative, and the outer membrane is present in the gram-negative, while outer membrane is absent in the gram-positive. So there are 3 layers in the gram-negative bacterial cell and the 2 layers in the gram-positive bacterial cells. Next point is the gram-positive bacterial cell wall. Now see what is the cell wall structure and the composition in the gram-positive bacterial cell. Gram-positive bacterial cells have much greater amount of peptidoglycan in their cell walls while the gram-positive and gram-negative cell walls have very less one. The cell wall of these bacteria consists about 40 to 80% of peptidoglycan of the dry weight of the cell wall. This peptidoglycan is of 40 or more layers in gram-positive bacteria. The cell wall measures about 30 to 80 nanometer in thickness. Teichoic acid and acid polysaccharide are mainly present in gram-positive bacteria and are found associated with the peptidoglycan by single terminal covalent bond. Teichoic acid is negatively charged. We have to remember this. Teichoic acid is negatively charged substituted polysaccharide polymer made of ribitol and glycerol residues joined through diphosphodiester bond. It constitutes a major surface antigen. It is a hydrophilic and its main function is to transport positively charged substances to the bacterial cell in the shortage of phosphorus. Teichoic acid first time discovered by the scientist Bacterial in 1933. Now let's see what is the cell wall structure of gram-negative bacterial cell. The envelope of this kind of bacteria is made up of two membranes and are separated by 100 angstrom space known as periplasmic region. It contains peptidoglycan layer. The outermost membrane is known as the cell wall, while the innermost membrane is known as cytoplasmic membrane. Peptidoglycan is of 5 to 10% of the dry weight of the cell wall. The outer membrane serves as a selective barrier for various external chemicals and enzymes that could damage the cells. Its structure is similar to the plasma membrane or cell membrane. The outer membrane is anchored to underlying peptidoglycan by means of Brown's lipoprotein. The membrane is bilayer structure consisting mainly of phospholipids protein and lipopolysaccharide structure. Now see, the gram-negative bacterial cell wall is three-layered structure, which the phospholipids are bilayer consisting both hydrophilic and hydrophobic ends. The walls contains 4 types of proteins components along with the major proteins known as lipoprotein. The lipopolysaccharide, which is called as LPS, has toxic properties and is also known as endotoxin. So lipopolysaccharide is a endotoxic in nature. It mainly causes the fever. It occurs only in the outer layer of the membrane and is composed of 3 covalently linked parts: lipid A, core polysaccharide, O antigens. Lipid A is firmly embedded in the membrane, core polysaccharide located at the membrane surface, and O antigen, which is mainly plays role as a endotoxin. The outer membrane is through impermeable to large molecules but can allow smaller molecules such as nucleosides, oligosaccharides, monosaccharides, peptides and amino acids. This is accomplished by means of channels in the special proteins called porins. So this is all about the gram-negative cell wall. So this is all about the gram-positive bacterial cell wall and gram-negative cell wall. Thanks a lot.